Hello, this is Yukio Nagasaki from University of Tsukuba, Japan. What I'd like to talk about today is a design of short-chain fatty acid-based self-assembling drug for alleviation of type 2 diabetes mellitus. This work have done by Babita here and Tajika here. Let me explain present status of drug development. As you know, more than 120 years, many types of drug have developed by organic synthesis. However, recently, no candidate for a leading drug is available which limit the organic synthesis development. 20 to 30 years ago, new drug based on protein started. Recently, many type of protein drug available. However, cost of these drug is increasing significantly, which cause another problem. We wanted to deliver cheap drug by a high performance nanocarrier. As you know, EPR effect accumulate nanocarrier into a target site. However, accumulation efficiency is few percent, which means more than 95% of nanocarrier spread to other organs. In addition, recent clinical trial shows leaking of drug is one of the other problem, which means even after accumulation of few percent of nanocarrier, which might be empty due to the leaking of drug during circulation, which causes limit of drug delivery system. So we wanted to develop the self-assembled drug. What is this? Creation of pharmacological activity that occur only after self-assembly, reduction of side effect, and avoidable with low molecular weight drug alone, cheap and safe material design technology. So today we focus on short-chain fatty acid. Let me briefly explain about short-chain fatty acid. As you know, indigestible vegetable cannot digest in intestine. However, the bacteria in colon digest them to form short-chain fatty acid like acetic acid, propionic acid, butyric acid. Recent studies showed, these kind of short-chain fatty acid have a versatile viable activity, pharmacological activity. This is why many people wanted to utilize them. So this is one of mechanisms which short-chain fatty acid interact with histone deacetylase or G protein coupled receptor, which control gene expression or expression of protein. Another paper shows, short-chain fatty acid interact with pancreas and control diabetes like this. So we wanted to use this kind of short-chain fatty acid. But what is problem? As you know, bad smell, bad odor of short-chain fatty acid, we cannot drink them. Rapid excretion due to the low molecular weight hydrophilic compound and adverse effect due to the acid nature. So the food industry recommend to eat, this kind of vegetable, supplementary industry prepare like this kind of supplement. But we, as a polymer chemist, we design new self-assembled drug based on short-chain fatty acid like this. Let me briefly explain. This is vinyl acetate hydrolyzed to vinyl alcohol. We learned this in high school in Japan. At the same time, the acetic acid liberate. We modified this reaction. We designed, polyethylene glycol polyvinyl short-chain fatty acid block copolymer, which because of the amphiphilic character, which formed the polymer micelle like this, we installed propionic acid, butyric acid as an ester linkage, and which can be cleaved by enzyme here. So after administration orally, which accumulate in intestine, hydrolysis by intestinal enzyme, blood uptake of ester, and distribution in organ. So this is not a drug delivery system. It is just nano-controlled release from intestine. Let me show you several results. We use, sample preferential test. We put, water bottle and sample bottle at the same gauge and confirm which one prefer by mouse. Sample was propionic acid, butyric acid, nanopropionic acid, nanobutyric acid here. You can see here the results. In case of nanobutyric acid, 75% the mouse prefer to drink sample. Only 25% they prefer. In case of nanopropionic acid, 50-50, and butyric acid and the propionic acid, less than 25%. I don't think, they confirm smell of sample, but I believe, acidic nature of these low molecular weight acid prevent, the uptake from them like this. So this is pharmacokinetics of our polymer. By iodine labeled sample, we installed iodine 125 at the end of polymer chain, and here radio labeling as a ester group here. You can see after oral administration, 24 hours after oral administration, the polymer here only locate in GI tract. No radioisotope observed in blood and liver, which means no internalization of polymer in bloodstream. In this case, when we are labeled as in a ester group, in addition to GI tract, radio labeling, radioactivity observed in all organs like this, which means after cleavage of ester group distribute in main organ like this. Next, we confirm therapeutic effect of diabetes mouse. We use, DB transgenic mouse, administer, short-chain fatty acid and nano short-chain fatty acid by free drink for several weeks. As a control, we use exenatide. This is commercially available GLP-1 receptor agonist. As you know, this is oligopeptide. We cannot administer orally. We administer this as a subcutaneous injection. So this is result, glucose tolerance test, which means we administer after 37-day treatment, we administer 2 grams of glucose, and after 1 hour, we confirm the glucose level in blood. The diabetes mouse shows like this. As compared to diabetes mouse, the nanobutyric acid treatment mouse decreased the glucose level like this. This is safe or better than commercially available exenatide, which is administered by subcutaneous injection. Nanopropionic acid works to some extent, but butyric acid better. Butyric acid, propionic acid did not work at all like this. So this is the beta cell Langerhans island. Due to the damage by diabetes, the size of Langerhans island decreased. So we confirm the size of Langerhans like this. In the case of, as compared to diabetes mouse, nanobutyric acid and exenatide keeps the large Langerhans island like this, which also evidence that they suppress damage of diabetes. So this is damage of intestine. As you know, the inflammation or damage of intestine decrease the length of villi we confirm the villi length. Like this is duodenum and column. As compared to the nanobutyric acid or nanopropionic acid, the low molecular weight butyric acid, propionic acid decrease the villi length. Exenatide also decrease the villi length, which is reported by other publication. Although this is injected subcutaneously, colon also shows similar effect. So this is probably due to the acidic nature of low molecular weight acid like this. So this is other damage of organ and blood. Due to the time limit, I skip most of them, but you can see spring weight increase in case of propionic acid, or platelet number increases propionic acid and exenatide. Most of data, the nanoparticle did not show, any damage to organ and blood. In conclusion, amphiphilic block copolymer having short chain fatty acid as a side chain of the hydrophobic segment was synthesized. The obtained block polymer forms, nanoparticle with several 10 nanometers. Nanoparticle accumulate in gastrointestinal tract by oral administration and hydrolyze, and then release short chain fatty acid to blood flow. Pre-oral administration of nanobutyric acid to diabetes model mouse showed no side effect and high diabetes treatment effect. Thank you very much for your kind attention.